Hello, everyone. My name is Rob Kozak. I'm a proud UBC alumnus and honored to serve as the Dean of the Faculty of Forestry. I'm really delighted to be your host for our virtual program this afternoon entitled The Carbon Flux of Life, featuring our very own Dr. Cindy Prescott. Before I begin, I would like to respectfully acknowledge that UBC's Vancouver Point Grey campus is situated on the traditional ancestral unceded territory of the Musqueam people, and that is where Cindy and I are speaking from today. So for millennia, these sites have been places of learning where cultures and histories and traditions are passed on from one generation to the next, and it's very much in that spirit that we're gathered here today. I'd also like to recognize that you are joining us from many places today, near and far, and I would encourage you to acknowledge the traditional owners and caretakers of those lands. So on behalf of the Faculty of Forestry, I'd like to extend a warm welcome to everyone joining us today, and thank you in advance for helping to create a respectful and inclusive environment for all of our speakers and audience members. This webinar is part of a successful series of conversations that we've hosted at the Faculty of Forestry. And today I'm really excited that we will be taking a deep dive into an incredible world of biodiversity that many of us take for granted or perhaps don't even consider. I'm of course referring to the complex world that exists below ground in forests and how organisms that live there are dependent on carbon fluxes produced by trees, which I guess we can add to the long list of benefits that trees provide our planet. And so by the end of this hour, we hope that you walk away with a whole new conception and appreciation of soil. And I can think of no better person to speak to this fascinating topic than my friend and colleague, Dr. Cindy Prescott. So just a quick bit of housekeeping before I pass the mic over to our moderator. If anyone is experiencing any audio or video issues, please just reach out to us via the chat feature for assistance, and we'll get on that right away. At this point, I'm honored to have the opportunity to introduce you to our moderator for today's webinar, another friend and colleague, Dr. Sally Aitken. Sally is a UBC alumna and professor with the Faculty of Forestry and former Associate Dean of Research. She's an internationally renowned scholar with a very long resume indeed. And notably in an era of climate change, she has been instrumental in reimagining the way we restock our forests with her innovative work in genomics and climate mapping technologies. She's also the coordinator for the BC Big Tree Registry, which documents the biggest trees in British Columbia. So thank you very much for joining us today, Sally. I'll now pass the screen off to you.
Thanks very much, Rob. It's a great pleasure to be here today and to lead this conversation with Cindy Prescott, who is really a very innovative scientist who has contributed so much to our understanding of processes, especially below ground in forests and in many ways. First of all, I'm coming to you from Quadra Island today, which is within the traditional territories of the Coast Salish peoples and the traditional lands of the Wiwaikai and Wiwaikum people. So after Cindy gives a presentation today, during the second half of the webinar, we'll be taking questions from our viewers. And we're using a platform called Slido to do this. And the instructions for Slido there they are, showing up on the slides. So your reminder email for the webinar included instructions on how to use Slido. It's very straightforward. A quick reminder of that, you can enter a question in Slido, or you can vote for a question from someone else that you think is particularly worthwhile. And I'll be drawing on those questions for our question and answer with Cindy after the seminar. And now for our topic today. More than half of the biodiversity in forests lives below ground, and the organisms that live in the soil depend on fluxes of carbon produced by trees and other plants. And Cindy is here today to talk with us about how surplus sugars that are exuded from trees nourish this underground world, and how we can apply that knowledge to support soil life within harvested areas. And the work and the concepts she'll be talking about today are really groundbreaking, and it's my great pleasure to turn the floor or turn the stage over to Cindy. Now, Cindy is a professor in the Department of Forest and Conservation Sciences at UBC. She joined the faculty in 1990, and she served as Associate Dean from 2003 to 2015. During part of that period, she was Associate Dean in 2 different faculties, and I have no idea how she managed to do that. Her research focuses on nutrient cycling and organic matter processes in forest ecosystems, including those factors that influence nutrient availability and carbon sequestration. And she's also studied the effects of forestry activities on soil processes, causes and corrections of nutrient deficiencies in regenerating forests, and the restoration of forest soils following disturbance. Cindy has received many remarkable recognitions of her contributions during her career. Just to mention a couple here, she received an honorary doctorate from the University of Helsinki, and that's a tremendous honor, and comes with a top hat and a sword. So just a little trivia there. Cindy will not be wearing her hat for her whole presentation, but there you go. And she also, it was just recently announced that Cindy will be receiving the IUFRO Scientific Achievement Award in June at the conference that occurs every 5 years. It'll be in Sweden this year. She also received the Canadian Forestry Scientific Achievement Award from the Canadian Institute of Forestry a few years back. So, and I'm a little sad to share with you, but excited for Cindy to say at the end of this month, after this very long and productive career, Cindy's retiring from UBC, but she will continue to share her knowledge as a professor emerita at UBC. So with that, welcome Cindy, and take it away.
Thanks very much for that kind introduction, Sally and Rob. I'll just get my screen sharing, hopefully correct. How are we doing? Okay, well, yes, thanks again. And also thanks again to our amazing alumni people for the invitation to talk in this really interesting series they're running this year. And of course, thanks to all of you for taking the time to tune in today or to the recording subsequently. So as Sally mentioned in the introduction, we now know that half of the biodiversity in a forest actually lives below ground. So soil is home to 90% of the fungi we know, 6% of the plant species, and yes, half of the plant is in the soil, and more than 50% of the bacteria taxa. Also an astonishing variety of animals, including protists, worms, and arthropods. And all of these are interacting in a complex dynamic food web that we're just starting to really understand. We know that together, these organisms break down all the organic matter the plants produce. They release the nutrients in forms that plants can use, thereby creating the foundation of the ecosystem. They determine whether carbon is sequestered for decades or centuries in the soil or released immediately as carbon dioxide back to the atmosphere. They also determine how much of the more potent greenhouse gases like methane and nitrous oxides are released to the atmosphere. So you'd think we'd know a lot about these organisms and the effects of our forestry practices on them, but in reality, we don't. The policies that govern forestry practices still largely treat soil as if it was an inert substrate rather than a living ecosystem. Policies are generally limited to preventing erosion and avoiding compaction, So don't lose it and don't squish it. And toss a log on top and the soil will be fine. But is it? So we could be forgiven for thinking this was sufficient when we thought that most soil organisms lived on the litter and dead organic matter that plants produce, which we did until recently. So there's a lot of this left after harvesting, as we know, especially here on the coast. And there's even more when you consider that there's even more debris left below ground as dead root systems and dead mycorrhizal fungi. But what if dead organic matter isn't the only fuel for below ground life? We now know that there's another flux of carbon straight from the leaves of trees to the below ground ecosystem. And I'll be explaining to you how we have come to know this. So about 20 years ago, in a Swedish boreal forest, the Höjbergs monitored CO2 release from the soil before and after girdling all of the trees in this plot, which prevented the movement of sugars from the leaves to the roots. They found that CO2 release from the soil plummeted within hours, and it indicated that more than half of the CO2 released below ground is derived from recent photosynthesis in the leaves and not from litter. Subsequent tracing studies with labeled carbon that's fed into the leaves either as labeled CO2 or injected directly into the stems of the trees as labeled glucose have been done, and they've been able to track the incorporation of this carbon into carbohydrates and the movement of this fixed carbon through the tree and into the roots and mycorrhizal fungi. And they found this happened within 2 days, it was already in the mycorrhizal fungi. And then within 2 to 6 days, it worked its way into the soil, into the soil microbes and chytreids, mites, collembola, all these mesofauna. And then it was actually in the predators, all the predatory mites, etc., within 6 days. So this straight flow of carbon from the leaves into the below ground ecosystem. And so now we know that living tree roots actually act as a conduit through which plant carbon feeds the below ground ecosystem. So how does this actually happen? It happens mostly through 3 pathways. First, on the left, you can see they're shown in blue. These are nematodes around the roots that suck this sugary solution directly out of the living roots. Or next to it, you can see the purple mycorrhizal fungi. Then they're not actually purple, and here you can see some collembola that are eating those. And what they usually do is they really focus on the really soluble labile material in there. Like they don't eat the skin of the fungus. They're really going for this recent photosynthate in there. And then finally, lining the surface of the roots and the fungi, we have all these bacteria that you can see as the little blue and pink specks on the far circle. And they consume all these sugars as they come out, and then they're grazed upon by amoeba and other sort of microfauna. And all of these fauna then are fed up through the food web, but because they're preyed on by bigger organisms. And so you have this movement right out of the plant root or the mycorrhizal fungus, right through this below ground food web. So that raises the next question of why are trees sending so much fixed carbon into the soil food web? And I've been looking into this for a few years now, and I'd like to tell you what I found out. And that trees, like all plants, even algae, have this peculiar physiological quirk that leaf growth and photosynthesis, they're both constrained by availabilities of nitrogen, phosphorus, and water, but they're separately constrained. And leaf growth is more sensitive to deficiencies than is photosynthesis. And so the growth of the leaf declines at a lesser or earlier stage of deficiency than does photosynthesis. So when plants are growing under mild to moderate deficiencies of nitrogen or phosphorus or water, which is pretty much every tree in British Columbia, the leaves will stop growing, but photosynthesis continues. And what happens in this situation, you can see in the diagram that these surplus photosynthates are produced. The same thing happens when we artificially elevate atmospheric concentrations of CO2. So the leaf cell ends up with surplus levels of carbohydrates, and they start to accumulate in the cells. And if they continue to accumulate, then there'll be less photosynthesis. It will be inhibited. But they don't tend to accumulate because they can be put into other compounds that don't require the nitrogen or phosphorus, such as starch or secondary metabolites, which will accumulate in the leaves. Or they can be loaded into the phloem and sent out, and they will head to all the active sinks in the plant. And so some will be used in the stem, some will go down to the roots. There they can be used for growth of the root if all of the other conditions allow, or they can be stored. But a lot of them will be transferred to mycorrhizal fungi, and this is a huge, usually active sink. Or they can be exuded out into the soil, and they can be exuded from the tip of the root, or they can be exuded by the mycorrhizal fungi. And so they find their way out into the soil, and they're accessible to the soil biota. And we know this happens with both bacteria and fungal hyphae, mycorrhizal fungal hyphae, because the surfaces of these tissues are lined with bacteria. And we know there's the rhizosphere, and now we know there's the hyphosphere surrounding all of the mycorrhizal hyphae, and that this is an active zone where surplus carbon from the plant is taken up and enters the below ground food web in the form of these bacteria. So this flux of labile plant carbon into the bacteria in the rhizosphere and hyphosphere is a major conduit into the soil. And we know, now that we know that it's coming out of the hyphae as well, this is a real game changer because there is so much fungal hyphae in soil. And here's a recent sort of average amount. In a cubic centimeter of soil, there's an average 7 centimeters of root tissue, but there's 2,000 centimeters of mycorrhizal hyphae. So that's essentially all of the soil would be receiving whatever's coming out of fungal hyphae. And we also know from research that's been done on soil organic matter formation in the last 2 decades that this is also a really important way through which soil carbon is replenished. So it doesn't just come from dead material, it comes from live material through the soil microbes, and their byproducts and their dead bodies are an important source of soil organic matter. So this flush of carbon actually helps to replenish even the more stable pools of soil carbon. And that's just one of the sinks for this surplus carbon that plants produce. Other ones include shaded understories plants, of course, that have access to nutrients, but they don't have access to the light to generate their own photosynthate. So they're able to tap into the living tree and its sugars through both mycorrhizal hyphae, of course, and through root grafts. And the same is true for living stumps. they're respiring, and so they're a carbon sink. And so as long as they're respiring, they will be pulling carbon in from the roots of the living trees. So it explains why we have these curious phenomena of these living stumps. Interestingly, another one was discovered by one of my graduate students, Adolfo Patti, in his work in dry IDF forests in British Columbia. And that's these huge wood ant nests, and you wonder, what are they living on? They're living on plant surplus carbon, which they collect from aphids on the needles of the trees that draw it out of the fir trees. So there's this really interesting flux of carbon, of labile carbon from the tree into the aphids. They give their honeydew to the ants. They take it back to their nest, and they use it to feed their young ones. And another student, Jocelyn Campbell, found this unusual abundance of non-photosynthetic lichens only under poplar trees in SBS forests. And she was able to attribute this to the poplars having extrafloral nectaries at the base of their leaves. And the purpose of these nectaries is actually to exude surplus sugar that these trees are producing. And they're able to get rid of it that way. And the lichens, these particular lichens, are only able to exist because of that little quirk. And also, the production of surplus carbon in forest peaks when the leaves stop growing in mid to late summer. So they stop growing, but they continue to photosynthesize. And thereafter, we see first the annual peak in stem growth as the surplus carbon starts to move. Then we see the peak in the growth of fine roots below ground, and then growth of mycorrhizal fungi. And then finally, the peak in production of their sporocarps or mushrooms in the fall. And so we know a lot more about this flux of labile carbon than we did. And I think we also need to now consider it in how we manage forests. So if this flux of labile carbon that comes out of the roots of living trees is indeed critical for sustaining below ground life and replenishing soil carbon, I think we probably need to leave more trees in cut blocks. So I've started looking at cut blocks differently than I used to, when I thought that the litter and slash was the source of all the energy and nutrients needed by the below ground organisms. But I look at them very differently now. So carbon tracing studies at the STEMS trial on Vancouver Island, as well as studies of the distribution of mycorrhizal fungi associated with individual trees, have demonstrated that the flux of labile carbon is greatest within 5 meters of a tree stem. And it's usually 0 by 10 meters from the stem. So one thing we could do is to minimize the portion of the cut block that's more than 10 meters from the stem of a living tree. And that would mean that most of the soil in that cut block had access to these sugars coming down out of the roots of trees and the associated mycorrhizal fungi. So this is an example of what we would be looking at. This is the dispersed retention plot at the STEMS trial on Vancouver Island. Here they left 40 trees per hectare, which is about 15 meters between stems. So here, if you looked at it from above and worked out how far 10 meters from a stem would be, you'd see that just about all, so much of that cut block would still be receiving this input from living tree roots. And so we would have, I think, a very dynamic below ground ecosystem in that area. In cut blocks, I think anything that's more than 10 meters from an edge or any sort of retention patch, I think, is probably very much depauperate in the soil biota relative to an uncut forest or a dispersed retention treatment. And I want to mention one more thing that the evidence is suggesting that we could do, which is to foster below ground biodiversity by encouraging above ground diversity, by increasing the number of plant species in our managed forests. And this is it's particularly important to have plants of different functional groups. Different types of trees have different communities of mycorrhizal fungi, and they also have different exudate profiles, we now know. Because experiments have made up exudate cocktails based on the composition of the exudates of different plant species. And they've applied these to the soil, and they've found that they can recreate the soil microbial community that they found beneath the plant community just by having those exudates, which means that to have the right level of below ground diversity, it requires that you have the right level of plant diversity. So another way we could foster soil biodiversity is by augmenting our conifer plantations with broadleaf trees or shrubs. And the most important ones seem to be the ones that have associative nitrogen fixing microbes, so such as red alder. These seem to be particularly important in this regard because they will exude both surplus carbon and surplus nitrogen that they have, because they make their own of both. And this seems to generate outsized increases in soil biodiversity when they're included in community of plants. And we also know that nitrogen fixing species are really important for rapidly regenerating soil organic matter and soil carbon stores. So just to finish off, why change how we manage forests? I think we all appreciate that managing our forests solely for the efficient production of timber is not enough in the 21st century, and that in our current era of record temperatures, extended droughts, insect and disease outbreaks, record wildfire years that seem to happen every year, floods, atmospheric rivers, our forests really need to be resilient. And I'll argue strongly that resilient forests begin with resilient soils. And this requires a diverse and fully functional below ground ecosystem. I'll just briefly put up some of the papers that I've been writing on this in the last few years. If you'd like to dig into this a little bit deeper. And I'll also put up my email address here. You're very welcome to contact me if you have any thoughts you'd like to share with me, or any questions that we don't cover in the discussion section. And thanks very much for your attention.
Thanks very much, Cindy. You've really given us a new perspective on what's going on in forests, and think about what we're walking on instead of just what we're looking up at.
I sure hope so.
Excellent reasons to diversify management practices and diversify the forests themselves. So before going to the Slido questions, I have a couple of questions for you.
Okay.
Doesn't come as a surprise. I have many questions for you, but some of them I think we're going to have to get together over a beverage and chat about. So
what a great idea.
Yeah. So first of all, you talk about leaving trees in harvested areas, more trees, so that we have more continuous root cover. And I know you've written a recent paper about that. That's very interesting. And I'm curious to what extent leaving shrubs after harvest, and you mentioned this a little bit, will help maintain soil biodiversity and health. And
Yeah, that's a really important point. Shrubs, especially if they're different functional groups from the trees, will be very helpful in that regard too. So as well as advance regeneration, of course. So it does sort of suggest that we also give a lot of consideration to that. And so not only should we be perhaps thinking about not compacting the soil, and that we should also be thinking about how valuable those plants that will survive the disturbance could be. And we also see that with fireweed and species like that in some situations will come back so quickly, because grasses, forbs, everything, they all exude carbon and particular complements of exudates into the soil. So that's another reason to take care of the soil and to take care of any plants that are there, as well as to leave the soil in a state that we know will invite a lot of plants to get going again. Yeah, that's an important point. Whatever ground vegetation is in there will also be really important.
Right. And many of those shrubs will also be producing fruits that further support biodiversity.
Absolutely. And this is just one part of the biodiversity, but it's the part that doesn't get much attention or thought. And partly because, until recently, we didn't know that much about it. We know there are a bunch of weird things down there, but we're now getting to a much better handle on who actually is down there, and starting to also get more information about what they do, which always just took so much work just to find out who was down there, and then to try to deduce from their mouth parts or whatever what they might be doing. So with each year, we're learning so much more about the actual biodiversity below ground, and it all makes me think that we really need to be thinking about this more when we're making decisions about that ecosystem.
Yeah, and a lot of the things you're talking about have a counterpart in some of the changes in agriculture that are going on around soils. We're not here to talk about those, but the recognition of the importance of soil health and different approaches to managing.
Absolutely. And the recognition that keeping that soil alive is so important. And that's the whole basis of regenerative agriculture, is trying to maintain this constant flux of these sugars and other exudates to keep that below ground ecosystem alive. And, forests are not anywhere near as devastating as arable farming. But in forests too, we should be thinking about it. Now that we've got this knowledge from regenerative agriculture that this really works, you can really keep a dynamic ecosystem down there if you always keep live plants growing and exuding into the soil. So don't ever have a bare soil. It's got to have plants. And that's going to give you all that below ground life, which is going to give you organic matter and start sequestering soil for us. Carbon. So I always think about soil, but carbon.
So we're getting some really good questions on Slido now, and I'm gonna and enthusiastic support from the audience for those. So right now, the top question is, how does soil biota recover after intense forest fires?
Now that's really interesting, because just because forest fires are natural, or we're contributing to them now, but just because it's a natural disturbance doesn't mean it is good for the below ground ecosystem. It is a really severe wildfire is devastating. And we know from how in some cases plant regeneration can be limited. It's the same below ground. You've changed so much about that surface soil that it is not a really good, it's not a good habitat anymore So we kind of thought, oh, well, it's natural, you know, and we were thinking that way for, I think, way too long. But if we're really thinking that we need this biodiversity and we need to foster it and we need to steward it, we need to think about really hot wildfires too. So that's another reason to have fires, like managed fires, that are not so hot. Because yeah, one of my students did a study with the effect of thinning the forest and then burning it in the slash piles, and he found the hotter it was, the more devastating it was for the soil biota. And so it takes so much longer to get that system regenerated if the soil has been that degraded. So we also have to get on to trying not to have our fires so severe. And there's so many other good reasons that we should be doing more controlled burns to try to keep it within the level of severity that allows that ecosystem to recover quite quickly from a mild burn.
So the next question that has the highest votes is, what are the three lowest hanging fruit in terms of changes to forest management that we can implement for what you're suggesting is missing in terms of the way we're managing forests?
Yeah, I think we need to backtrack to where we were at the start of the new millennium, where we were seriously thinking about alternative silviculture systems. And we'd done all those trials in the 90s, and we had variable retention harvesting brought in. And I think most people know we have slid back since then. Because when we had variable retention harvesting, what we meant by that was either having dispersed retention, and so having quite a few trees. We're not talking about leaving 5% of the trees in these, you know, these two stems, like I showed the photo of a recent cut up near my place on the Sunshine Coast. We're talking about a significant number of living stems left. And whether that's, you know, group retention is a no-brainer because you're leaving a chunk of the forest, which is fine. But every bit of that clear cut that's more than 10 meters from a living stem, I think is depauperate in its biodiversity. Above ground, yes. And now we know especially below ground. And I think we need to really look at those gaps that we're leaving and think, can we really not do better than this? You know, just to have a chunk of trees left in the corner and then clear cut the rest. That's not what we were talking about in the 90s, and that's not what we meant by variable retention. We need to bring that thinking back, that we're really talking about leaving a significant number of trees. 30% of the trees, basically you got a pretty good ecosystem there. If they blow down, they blow down. But that's no excuse to not leave them. So harvesting, I think we need to think more about and get back to where we were. And then the diversity. I mean, it's a kind of a knee-jerk reaction to think diversity is good, but the question always is, what is it good for? And we need to have good reasons for diversity. And I think we now have them in the last 2 decades since I've been putting all this together in the last few years. We have very compelling evidence for the importance of plant diversity, for below ground diversity. And we have compelling evidence for the importance of below ground diversity in making a functional ecosystem, for all the reasons that we need now to, you know, to have able to withstand all of these stresses that have come with climate change. So I think we really need to think about the wisdom of either monoculture or all conifer plantations. Those other species, especially if they come in there on their own. Let's use that. And especially alder, we have this plant that just comes back, and it's fantastic for putting nitrogen into the soil, and the rate at which that builds soil organic matter and soil carbon. That's a thing to have if you've got a disturbed site so that needs restoration. Get a nitrogen fixing shrub in there. But if you only have them, then you've got all sorts of other problems with nitrate leaching and pH and acidity and all that. So you need either even just 5 years of them, or to have a mixture that includes them. So there's just so many reasons to have more plant species. And we are so much smarter than I think our forestry practices give us credit for. I mean, we can manage mixed forests.
Yeah, I couldn't agree with you more. And the stands that are planted still get a lot of natural regen from various species. I think the average in one study was at least 500 stems per hectare, you know, and the old school view is those are the ones you want to take those out because you just want to, you know, manage those trees you planted. But that,
yeah, I mean,
in uncertain times, you know,
yeah, when the only thing that mattered was growth and yield of the crop tree, then that made sense. They're competitors. They're going to reduce in that immediate span of time. They're going to reduce the growth of that crop tree, but they're collaborators. If you think about it as an ecosystem, they're collaborating to make that a really functional, dynamic, alive ecosystem, both above ground and below ground. So maybe this is more like the First Nations would think about different species, you know, what they're useful for and such, but maybe they're actually collaborators and not just competitors.
we also don't know which species is going to be the next one to be hit with a particular insect or disease that
Exactly.
flares up. So
as far as I've been told, it sounds like the fungi and the insects are just queuing up.
Yeah.
Who's going to be the next big one?
So we've just got a lot of great questions here. So the next one I'd like to target, because it related to something you were just talking about, is have there been investigations of the diversity of soil biota along transects from forest into clear cuts?
Not nearly enough. We have done for mycorrhizal fungi and for microbial biomass, but there's not nearly enough. When I start poking around in the literature, well, not poking around, digging as deeply as I possibly could to find out what evidence we have about this, there's not nearly enough. And the reason was it used to take so long to extract all these organisms and then to identify it. I mean, it was just, it would be murderous to grad students having to do that. And I salute all of those who did, because it was so hard. But now there's all these omics that are making it possible to at least find out what is there and some indication of their abundance. So I think the time is right now to start really digging in and finding out what the diversity levels look like with distance from the edges and how that relates to the root system. Some of our students were doing it with labeled carbon and tracking into the microbes, but that was just, at that time, it was just with broad groups of microorganisms. But so much is possible now with the DNA metabarcoding, which you'll know 100 times more about than I do, Sally, so I'm not going to say any more about it, but we have tools like that now that can really give us an indication of diversity. And this study I mentioned right at the start that was able to actually get some sort of numbers for something that we've just never had numbers. And then when you had numbers, the numbers were just so incredible. They were just mind-blowing. And that coupled with the carbon tracing, I think those were real game changers that just showed us how dynamic that is. So I would say the time is now to learn this. And I'd love to see that sort of research in British Columbia. And we've got so many forest types, so you've got all these situations, but at least for some of the forest types, just to find out if the evidence I've been able to find from these other avenues of inquiry are correct, and that there is so much going on down there, and it is so dependent on living trees and keeping that system alive, even while we're using the forest for other purposes.
Yeah, absolutely. Just one comment, there's been a request for the literature shared out, that the literature that Cindy's talked about be shared out after the presentation. And I'll try to get a link to Cindy's Google Scholar page in the chat, but also I think that Michelle Lindsay has the capacity to send out the list of publications to everybody who's in attendance. So I think we can quite easily follow up on that request. And there are some excellent recent papers that Cindy has written that really point back up what she's saying with all the science on the topic and translate that into forest management impacts.
Oh, I should also mention that all the papers are open access as well. So everyone will be able to find them just by searching for me, and then you'll have full access to them.
Google Scholar is a wonderful way to find scientific literature, and you will be able to access it directly from that if it is open access. And if not, contact Cindy, and she can, well, she just said they're open access, but for any of the faculty members, contact us if there's a paper that is behind a paywall that you can't access, and we're always happy to send copies of our work. So
delighted, I would say.
Delighted there's interest. So I'm hoping to get to two last questions here from Slido. The first one from Barbara Happy is, at what point in the regeneration phase do you start seeing significant carbon flux impacts from young stands?
So I would say by the time you're at crown closure, there should be significant flux below ground. So you've got to build, it also depends on how many year classes of needles the species retains. Because what we've found is in our species trials is that if a species like spruce or fir, true firs that can have 20-year classes of needles, they take a long time to start influencing the soil, producing litter and such, but we don't know how long they take to start sending surplus carbon below ground. So it will depend a lot on the growth rate of the species. But I would think, especially after full leaf expansion in midsummer, that would be the time that we should start looking for it. And you could get an indication by, if you're starting to see the mushrooms of ectomycorrhizal fungi in the fall, that would suggest to me that you've got a significant flux happening below ground, and you've got enough roots and you've got enough mycorrhizal fungi established that they're able to funnel that late season carbon below ground. And at that point, if you're seeing that, I would be confident that it is now coming out of the hyphae into the hyphosphere, and so into the ecosystem. So I would guess crown closure would be the time that it's up and running in terms of this carbon flux. But then, of course, it doesn't have to be just your conifer crop crown closing. As soon as you've got the other plants, shrubs especially, in there doing things, then you've got those coming down as well. I have no actual evidence from studies that have looked at that question, because we are just so far behind what is now possible in terms of quantifying this and finding out, especially in our forests in British Columbia, how big is this flux, and questions like that of when it starts leading to the recovery of that below ground ecosystem.
Soil carbon is such a substantial portion of the total carbon pool of forests, and yet we still tend to think of the above ground portion alone and know so little about how much is below ground.
Yeah, and the soil carbon can be a very, just a slow loss. And so, you know, with all that slash in that, if you just measure total carbon, you know, there's going to be heaps of it because you got all that dead stuff below ground. But this labile flux is what is stopped when you take all the trees out. And we don't, and that's the stuff that has to be continuously replenished. It is the more persistent, especially if it's in contact with minerals in the mineral soil. But still, if it's slowly leaking, then it has to be being replenished, or those stocks are going to plummet. And so that's why I think we tend to have this kind of long-term loss of soil carbon while the system is regaining this other pipeline and having the carbon replenished through that. So as an indicator of what you would see above ground, I would say, if we're assuming that in most of our forests, if you have closed canopy, you also have essentially closed root. If you include the hyposphere in there, it's starting to fill in. And so that would be my guess for what you look for, in addition to the fruiting bodies of the ectomycorrhizal fungi. They're abundant, should tell you how much is being replenished below ground.
And they also tell you what age of forest to get out in to do some mushrooming. Yeah, okay, that's great. With that, given the time, I think that there was one more question about impacts in different BEC zones. That's a big topic, but I want to make sure we have time for a few last comments from you. So I'll turn it over to you, and you can choose which of those you will tackle.
Yeah, for BEC zones, that's the one of the main things I would say is we need so much research now on this, because the rest of the world has been carrying on and finding out a lot about forests, and all the things that I've described to you today. When I was here first in the 1990s and we were doing all this great silvicultural systems research and that, we didn't know any of this. So there is so much opportunity now to know more about the below ground ecosystem and its relationship with the living plants, that I think we really need to start asking these questions and trying to quantify these things in our forests. We have such an amazing diversity of forests, and as far as life below ground, we know so little about it. I think we now have the tools, I think, that we can find out. Sadly, I'm not going to be one of the ones doing it, because I'm hoping to minimize the amount of time I spend standing out in the rain or the snow, collecting soil from here on. But I hope others will be inspired by what is now possible to get this back on the agenda and say, let's find out about this. Let's find out if Prescott was just saying a bunch of crazy stuff, or if she might be right about this.
Yeah. Well, that's one of the marks of a good scientist, or a great scientist is recognizing all the things that we don't know, and all the things we need to keep on investigating. And I think you've stirred up this field considerably with the work you've already done. And today's seminar has been a real eye-opener, I'm sure, for many people. I've been following your work closely and really enjoyed seeing what's coming out. It's really opening a lot of new avenues. Seems to me that what it suggests is that we need more soil scientists in our faculty. But
low ground ecosystem. So that's what I want.
Ground ecologists, yeah.
Don't just see soil, good as it is.
No.
That's an ecosystem
Good
down
point.
there.
Below ground ecologists. So thanks so much for what you've shared with us today. I want to thank everybody for joining us and for asking great questions on Slido. The references from today will be sent out with a link to the recorded webinar. And as always, if you want to learn more about the Faculty of Forestry, you can visit us at www.forestry.ubc.ca. And we will be sharing the recording of this program quite soon. We wish everyone a happy and safe and healthy holiday. Thanks very much for joining us, and have a great rest of your day.